Happy month of May, everybody. In Sweden, it doesn't really look like May yet, but hopefully soon. If you are joining us from some other place in the world or watching this afterwards, I hope you're having a really nice day. And today we have, like we've been doing this 2025, presenting presentations by our PhD students at the center. All of them have already passed their halfway point. A lot of things coming up, results, projects being developed. And today we have Patrícia Coehlo and we have Ottilia Andersson. We're going to start with Patrícia and then we'll have a little of a leg stretcher, 5-minute break, and then we'll continue with Ottilia. So it's my pleasure to introduce our PhD student, Ottilia Coehlo, from the Department of Biochemistry and Microbiology. And she is going to talk to us about biofilms, those pesky forms of life that bacteria can have that are actually very important in the clinics, and in particular, working with biofilms in Klebsiella. So, Patricia, the floor is yours. Just some housekeeping things. We are recording the webinar, so you are, of course, welcome to open up your mic and your camera later for the Q&A for both. But if you don't want to be on camera or you don't want your voice to be recorded, you can always write the questions in the chat, and I will be able to give it to the speaker so we can have a little bit of a discussion about their work. Patricia, the floor, digital floor, is yours. Looking forward to see how your projects are going.
Can you guys see my screen?
Yes, correct.
Let me just move this thing. Okay. So thank you, Eva, for the introduction. So, wait, I want the laser. Yes. So I'm doing a PhD in Linus Undergrand's group, and I'm studying biofilms formed by Klebsiella pneumoniae. And today I would like to present some of my work, which is about the evolution of Klebsiella pneumoniae biofilms and how it affects the efficacy of antibiotic treatment. So first, I would like to explain a little bit what a biofilm is, in case you guys don't know. So by definition, a biofilm is a polymicrobial community, and they can adhere to each other. So these cells can adhere to each other and or to a surface. And usually they are coated in a self or host-derived matrix. So there is this sort of slimy gel consistency substance, and this is what we call the biofilm matrix that encases the cell within the biofilm. So typically, you look at a biofilm in many papers, books, whatever, and you see a structure like this, but biofilms are much more complex than that. So usually biofilms look more like this. So there's cellular components, and you can have different cellular populations, so to say. You can have like normal bacterium. In some instances, you might have resistant bacteria that produce antibiotic-degrading enzymes or other resistance genes. There can also be dormant bacterium within the biofilm, and I'll get back to that a bit later. And there could also be dead bacterium, and these cell debris also work as building blocks for bacteria that are growing within the biofilm. Apart from this, there's also non-cellular components. So within the matrix, you might have carbohydrates, extracellular DNA, proteins, and lipids, and this constitutes the extracellular matrix. And if there are resistant populations within the biofilm, you can also have antibiotic-degrading enzymes within the biofilm matrix. When it comes to the steps of biofilm formation, biofilm formation is a very dynamic process. So biofilms are in constant production or formation and degradation, but it mainly consists of 4 steps. So first is attachment. So the cells adhere to each other or to a surface. And at this point, the attachment is reversible. So at any point, they can detach and continue in a free lifestyle, so to say. The second step is colonization. So they continue binding to each other and they start producing the biofilm matrix. And at this point, the adherence between the cells becomes irreversible. So they can no longer detach from each other. And at this point, the biofilm forms what we call a microcolony. So it's like a small baby biofilm, so to say. Then the biofilm grows, bacteria multiply, and there's the emergence of different subpopulations within the biofilm. And this we call the maturation step. And finally, we have the dispersion. So there can either be dispersion of free cells that will colonize other places, or there can also be release of chunks of the biofilm to continue biofilm growth somewhere else. But how or why are biofilms important in the clinic? So most people don't actually know this, but about 65 to 80% of all bacterial infections are related to biofilm formation. And in fact, bacteria, even in nature, about 90 to 95%, they exist within biofilms. So I think it's safe to say that bacteria choose to live within the biofilm and it's more favorable for their survival. In the body, biofilms can manifest in many different ways. Some are infectious, some are not. So for instance, on your teeth, you might have dental plaque, and usually these biofilms are relatively harmless if people have proper oral hygiene. There can also be biofilm formation in contact lenses if they are not properly taken care of and cleaned. In most serious cases, there can be biofilm formation in any foreign object that you introduce in the body. So for instance, artificial heart valves, hip replacements or knee replacements, so any sort of also implants that are introduced in the body, and finally catheters, and this is highly associated with urinary tract infections. But biofilms form also in biological surfaces, not only medical devices. So for instance, there can be biofilm formation in the epithelium of the alveola in the lungs, in the intestine, and there's some studies showing associations between biofilm formation in the intestine and cancer development. And finally, there can also be biofilm formation in chronic wounds, and this makes these wounds really hard to treat, and they persist, and that's why they're called chronic wounds. Biofilms are serious and they are a big problem because not only do they resist the host immune system, but they are also very tolerant to antibiotic treatment. And why did we choose to study Klebsiella pneumoniae biofilms? So Klebsiella is a gram-negative bacterium that is naturally present in the microbiota, and it is an opportunistic pathogen. So it mainly infects immunocompromised individuals. However, between 2005 and 2010, there was a Klebsiella pneumonia outbreak at the Uppsala University Hospital, where it's estimated that about 320 patients were either colonized or infected by the same multi-resistant clone. And there was a study done in our group as well, where they looked at the in vivo evolution of this clone within the host during the outbreak. And they saw that this clone evolved and adapted changes in genes often related to capsule, iron acquisition and metabolism, cyclic GMP synthesis, and Fimbria as well. Furthermore, they also saw that these changes led to a chronic and not acutely virulent states, and these mutants also had an increased biofilm formation capacity. So the question is, would we be able to see this also in vitro? So would we be able to see the same evolutionary trajectory in an in vitro setting? So that's where the project starts. We chose 3 clinical isolates to evolve. So we have a respiratory isolate here, 1 isolate from a urinary tract infection, and the isolate from the outbreak. And we evolved these 3 parental strains in different surfaces. So in polystyrene, which is normal 96 well plates, and also different types of pegs. So we have this device here that was developed in our lab as well. And we have individual pegs that are inserted in a multi-titer plate. And the cool thing about these pegs is that we can coat them with different materials. And in this study, we actually used uncoated pegs. So this is just a normal high temperature resin. And we also coated the pegs with silicone to mimic the medical devices. And even in the pegs are coated with silicone, we also coated them with fibrinogen because fibrinogen is usually released by the host when there's an insertion of a catheter during a urinary tract infection. For the evolution, the biofilms were formed for 48 hours on these pegs. After 48 hours of incubation, they were dispersed by vortexing. Part of the population was kept for re-inoculation and continue the cycle. And the other part of the population was analyzed for CFU counts and biofilm formation capacity by crystal violet staining, and also whole genome sequencing. And this was done over the course of several cycles. So the main two observations that we did from this first step was that there is a gradual increase in biofilm formation capacity, and this is very rapidly selected as well. So within a couple of cycles, we picked up mutants that are very good biofilm formers, and this also led to an accumulation of biofilm biomass. And when looking at the whole genome sequencing results, and here we have three different graphs for each three of the parental strains that were involved. So we have the respiratory one here, the UTI parental here, and the outbreak. We saw that some targets were repeatedly selected, and they mostly consisted in three different groups. Capsule, so we have, for instance, a WZC gene here, here, and here. So there was common for the three families. Type 3 fimbria, for instance, the MRKD gene here and here as well, and the cyclic GMP regulation. And I'll get back to this a bit later. So these three main targets were repeatedly selected, regardless of the parental strain that was evolved from. Now looking at the mutants in more detail, starting with the capsule, we saw that this altered capsule production was due to mutations in WZC. So we have a WZC protein here, and it is responsible for the polymerization and export of capsule polysaccharides to the outside of the cell. And we saw that these mutants have an increased capsule production, usually overproduction of capsule. But even though they overproduce capsule, there's distortions in the cell envelope and less defined LPS as well. So if you look here, you don't have a very well-structured capsule. You have more of like a blur of capsule polysaccharides surrounding the cell. There's also shedding from the cell surface. So if you look here, you have polysaccharides that are shedding from the cell. And there was also accumulation in the periplasm. So if we go back to this figure, the periplasm is the space between the two membranes in gram-negative bacteria. So it seems that the capsule polysaccharides are partially produced and then they get trapped in the periplasm and they are not exported into the outside of the cell where they should be. Now moving on to the Fimbria mutants, we saw that these mutants were very good biofilm formers, and they had mutations in type 3 Fimbria and mainly on the MRKD gene. So this is a type 3 Fimbria here, and it is responsible by binding to different surfaces, and the MRKD is the tip here. We don't know exactly what is the effect of this mutation yet, but we think that there is a changed or deleted tip subunit. And you might think, okay, if you don't have the tip subunit, they will not be able to bind to surfaces as well, but it's actually the opposite. So they lose the tip subunit and they bind even better. And the reason for it is that the MRKD is responsible by binding to biotic surfaces, so biological surfaces like epitheliums and so on. But the MRKA, which is these subunits here, can also bind abiotic surfaces, so artificial surfaces like medical devices, implants, and so on. So it seems that if we are evolving these mutants in silicone-coated pegs or in plastic, they lose this tip to bind better to these types of surfaces that are not biological. And we can see here, these are results from the crystal violet staining, and that shows how much biomass is the biofilm producing in total. And the bluer, or the more blue it is, the more strong is their biofilm formation capacity. And we see that in multiple different surfaces. So we have silicone here, silicone and fibrinogen, and also polystyrene. And in fact, these biofilms were even better at binding to silicone and fibrinogen, and this happens even at early steps of biofilm formation. So in this graph, we have biofilm formation within 3 hours of incubation. So we can see that even after just 3 hours, some of the mutants are much better than the parental strain at binding and forming biofilm. And the last group of mutations that we saw were involved in the cyclic DGMP turnover. So cyclic DGMP is a second messenger. and it controls many bacterial regulatory pathways, one of them being biofilm formation. And there's still much that is not known about exactly how the cyclic GMP control biofilm formation, but the rule of thumb is at early steps of biofilm formation, or we don't even have a biofilm here yet. So while bacteria are still in planktonic phase, so while they're still floating, there's low levels of cyclic GMP inside the cell. Then when the bacteria start to bind, there's a switch in genetic expression and they form the microcolony. And at this point, the cyclic GMP levels start to increase and they form a more robust biofilm. And then when it comes the time to disperse the biofilm, the cyclic GMP levels go down again. So high cyclic GMP levels are associated with strong biofilm formation, while low cyclic GMP levels are associated with no biofilm formation and more mobile lifestyle. And it is known that cyclic GMP actually regulates the production of fimbrial adhesins and also polysaccharides that compose the extracellular matrix. So within this group of mutations, we saw that a lot of these strains had mutations in the MRKJ gene. So if we go back to the previous figure that I showed you about the type 3 fimbria here, we have the synthesis of cyclic GMP. So initially we have GTP that is converted to cyclic GMP. When there's high concentrations of cyclic GMP in the cell, it binds to the transcription regulator MRKH, which activates the expression of the MRK operon. And you can see that this operon actually has all the building blocks necessary for the production and assembly of MRKD fimbria. So it will regulate biofilm formation by increasing the expression of type 3 fimbria, so bacteria will bind better to surfaces and form stronger biofilms. However, we don't know exactly what is the effect of this mutation on the MRKJ. And initially we thought that it would perhaps be a loss of function or at least decreased activity of this protein. So the hypothesis was that if there's a decreased activity of the MRKJ, which degrades cyclic GMP, the levels of cyclic GMP within the cell will be higher, and therefore the expression of this operon will be higher as well. So we looked at the expression of MRKA by qPCR, and we saw that most mutations increased MRKA expression. However, this was very strain dependent and also growth state dependent. So we couldn't exactly see a correlation between the mutations and their effect on MRKA expression, and also a correlation between how strong the biofilms are and the MRKA expression. So we would think that the more biofilm these strains are producing, the higher expression of MRKA would be, and we didn't exactly see that. So we don't have an exact trend that could explain why these bacteria form biofilm so well. So we concluded that differences in MRKA expression alone cannot fully explain the biofilm formation capacity of these strains. However, we also saw that these mutants, or some of these mutants, produced a very prominent extracellular matrix. So if you look at this figure here, you have these strands of polysaccharides around the cell. And it is thought that MRKJ and cyclic GMP could regulate the expression of these capsule polysaccharides. And some studies indicate that cellulose can actually be a key component of the capsule in these mutants, or in Klebsiella pneumoniae biofilms. So we also looked into cellulose production by doing qPCR on the BCSA gene, and we didn't see a lot of changes in the expression of this gene for most mutants. And again, it was strain dependent and also background dependent, so the family that the mutant is derived from. However, if we treat these biofilms with cellulase, which is an enzyme that degrades cellulose, we see that some of these mutants actually are very sensitive to it, and the biofilm formed by these mutants is completely eradicated. So it seems that cellulose is important, but the regulation of cellulose production is not regulated at a transcriptional level, but more at a post-transcriptional level, because we didn't see any differences in BCSA expression. But like I said, during the introduction, we had the in vivo studies that showed that within the host, Klebsiella evolves toward a non-virulent state. So we wanted to check that also in the in vitro evolution of our strains. So we exposed these biofilms to human serum, and we also checked their effect on Galeria melonella larvae, if they could kill the larvae better or not, depending on the mutation that they had. So we saw that by exposing these strains to serum, the WZC mutants were extremely serum sensitive. So you would think that by overproducing a capsule, you might have more protection from human serum and the complement system, but it was the other way around. So it kind of confirms that perhaps they are overproducing capsule, but the capsule loses its structure. So the complement system can actually affect the cells a bit better. But on the other end, the MRKD and MRKJ mutants showed a better survival in human serum. So they were very much tolerant to exposure to human serum. But in Galeria melonella, the results were a bit more non-conclusive. So there was a strong strain-dependent effect on virulence. We couldn't see correlations between mutations and strains and how they affect survivability of Galeria melonella. So for instance, we saw that some WZC mutations decreased killing of larvae. So for instance, here we have in blue the WZC mutations. Here the larvae are pretty much okay. So the WZC mutant is not doing very much. However, here the WZC is extremely dangerous against the larvae. So they are completely dead after just 24 hours of injection. So we don't see a correlation between the mutants and the mutations that they have and how they affect the survivability of Galeria melonella. So far, we show that Klebsiella can rapidly evolve towards a stronger biofilm formation capacity phenotype, both in vitro and in the different abiotic surfaces. Vital cellular structures and processes are the main mutational targets, and they are repeatedly selected. And there is a strong strain-specific bias on how the phenotypic effects are. So we don't see, again, a correlation between mutations and the final outcome in the phenotype. So moving on, we know that there are mutants that are rapidly evolved and they adapt a more or a stronger biofilm formation capacity. We know that three main targets are repeatedly selected, so capsule, type 3 fimbria, and cyclic GMP turnover. So now the obvious question is, how would this affect antibiotic susceptibility? So that's what we looked into afterwards. I chose one representative mutant from each of these targets and one or two of the parental strains that we studied before. So we have the respiratory one here and the one from the UTI infection, and a WZC, an MRKD, and an MRKJ mutant. And we exposed them to antibiotics of different classes so we can get a broader picture of how these biofilms behave following antibiotic treatment. The setup was more or less the same from before, so we are still using silicone-coated pegs. The biofilms are formed for 48 hours, and then they are exposed to antibiotics at different concentrations. Then we assess two different things, either viability by doing CFU counts or biofilm biomass by doing crystal violet staining. And I know this table is big and you are not supposed to check the numbers, but here I just want to emphasize two things. We have the MBC, which is the minimum bactericidal concentration in liquid, and then the MBEC, which is the minimum biofilm eradication concentration. And we can see that by simply having biofilm formation, these concentrations are much higher than the MBC. And in most cases, we couldn't even calculate an MBC, which means that the actual value is even higher than the range that we used here. So these biofilms are extremely difficult to eradicate, even at high concentrations. We could determined some values here, for instance, for gentamicin. So it seems that gentamicin had some effect on these biofilms, but these concentrations are extremely high and they are not relevant in the clinics because, especially gentamicin, which has really bad side effects. So the concentrations used to treat the patient would not have any effect on these biofilms. But we also looked at how the biofilms behave within a range of concentrations, so from 1/8 to 8 times the MIC. And in black, we have the parental strains. In beige, we have the WZC mutants. And we have the MRKD mutants here in gray, and also the MRKG mutants in blue. So we can see that overall, the mature biofilms, so biofilms formed for 48 hours, are fairly tolerant to the majority of antibiotics tested. So at best, we could have a 2 to 3 log decrease in biofilm or in cell viability. And then there was like a stagnant step. So we couldn't even get lower than this. So most of the antibiotics had no effect on the biofilms, regardless of the strain that we were testing, so the parentals or the mutants. Here we have the crystal violet results, while these were CFU. So we can see that the capsule mutants, so these ones here in beige, were more sensitive in most of the cases, even than the parental strain. And if you look back into the serum killing, I said that the WZC mutants were extremely sensitive to serum. So it seems that we are having a pattern here. WZC mutants are not only sensitive to serum, but they're also more sensitive to antibiotic treatment. But on the other hand, the MRKD and MRKG mutants are very much unaffected by antibiotic treatment, even at high concentrations. So when we think about how biofilms are tolerant to antibiotic treatment, even when the strains are not exactly resistant to antibiotics, there are different theories that can explain this. So for instance, there's a decreased diffusion. So it is thought that the antibiotic cannot penetrate the matrix, or if it can penetrate the matrix, it will be at very low concentrations, and therefore the biofilm will not be affected by the antibiotics. Then, if we think about a biofilm as a three-dimensional structure, the cells here in the core will be deprived of oxygen and nutrients, so they will become dormant and have a lower metabolic activity. So antibiotics that rely on bacteria that are growing to have an effect will no longer have an effect on the biofilms. And then, if the antibiotic can penetrate the matrix, there might be components within the matrix that inactivate the antibiotic. So either the proteins or the lipids that are within the matrix bind the antibiotic and sort of sequester it, or there's some pH that render the antibiotic inactive, and so on. So we went to look at this, and we decided to grow the biofilms as we had done, but instead of exposing the biofilms intact to antibiotics, we dispersed it by vortexing. So by doing this, we sort of got rid of the structure of the biofilm, and we had the same populations that we have in the biofilm, but dispersed. So we no longer have the structure of the biofilm to protect against antibiotic treatment. And we saw that for some antibiotics, and we have here this graph where the sort of full dots are the bacteria exposed in medium. The empty dots are the bacteria exposed in just PBS, and then the empty dots with a dot inside were disrupted before antibiotic treatment. And for some antibiotics, like ciprofloxacin, gentamicin, and enoxaparin, this actually worked. So by getting rid of the structure of the biofilm, these antibiotics were actually able to kill the bacteria, and sometimes even completely eradicating the biofilm. But for other antibiotics, we didn't see that. For instance, for cefotaxime, no matter what we did, the susceptibility of these strains was very much the same, which indicates that in this case, it's not the structure of the biofilm that is protecting the cells, but it could be different subpopulations within the biofilm. So for instance, dormant cells. And to account for that, I don't show these results here because we are still validating them, but we try to kickstart the bacteria prior to antibiotic treatment. So we did that with mediums to sort of wake them up if they are dormant. And then we expose them to antibiotics after waking them up with medium. And it's actually worked. So for cefotaxime in specific, by doing this, we could actually lead to complete eradication of the biofilm after treatment with cefotaxime, sorry. And one interesting observation that we made was also that some antibiotics induce biofilm formation at sub-inhibitory concentrations. So for instance, here, if we have a more gray color, there is an increased biofilm formation. And we can see that cefotaxime is one of them, and also colistin for some of the strains. And in these cases, there was only an increased biofilm biomass production, but we didn't see an increase in CFUs. So it seems that this effect, it's only by inducing the production of the extracellular matrix. So we looked into matrix components. Here we have total polysaccharides and cellulose. And we can see that for some strains here, there is a drastic increase in polysaccharides and cellulose production following treatment with some antibiotics at sub-MIC, but we don't see the same effect, for instance, for extracellular DNA or proteins. So it seems that this effect is strictly on polysaccharides and cellulose in specific. And this confirms that cellulose is potentially a key component of the biofilm matrix produced by the Klebsiella pneumonia. So we saw that standard antibiotic treatment fails to eradicate biofilms in vitro, and capsule mutants are sensitive to antibiotic exposure to some extent. And we see the same observation with serum killing. Furthermore, changes in fimbrial adhesion structures and also in cyclic GMP turnover result in increased antibiotic tolerance. And sublethal concentrations of antibiotics can actually induce biofilm formation by inducing matrix overproduction. But why is this relevant? So if we think about the in vivo study that we talked about in the beginning, so within the host, we saw that this clone evolved by acquiring changes in capsule, iron metabolism, cyclic GMP, and fimbria, leading to chronic, not acutely virulent states and increased biofilm formation capacity. But if we do the in vitro studies, we saw that these strains sort of replicate the adaptive changes or some of the adaptive changes observed in vivo. So we still have the same changes in capsule, cyclic GMP, and fimbria, which led also to an increased biofilm formation capacity and decreased antibiotic susceptibility. So it's safe to say that if we see these changes in vitro and decreased antibiotic susceptibility in vitro, we will also be able to see it in vivo within the host. So we might have to rethink how we treat these infections because most studies or most clinical studies are done in liquid cultures, so planktonic bacteria, which doesn't reflect how bacteria actually live not only in nature but also within the host. So they choose to live in biofilms, so we should think about how to treat these bacteria that live in biofilms, not in liquid cultures. So I would like to thank my group and Thomas Tengden's group as well, who is my co-supervisor, and of course, Uppsala Antibiotic Centers and my colleagues at D73 and D74 corridors. And thank you all for listening. Yes.
Thank you so much, Patricia. If any of you have any questions, you're getting some applauses, digital applauses. If you guys have any questions, you are welcome to put up your hand and you can open up mic and camera if you want to ask her directly or write on the chat. We have some minutes for some questions. Whoa, Patricia, so much data, so many experiments.
And I omitted a lot of it.
I can say that because I work in a similar type of project and lab, so I know how much time and work it goes into doing all these mutants and all these experiments. I did write a couple of things here regarding, I'm thinking more of the clinical aspect of this. So you used heavily synthetic materials like these silicone coatings and stuff like that. One of the materials wasn't more biological base or not? They were all artificial.
So for some of the experiments, we also coated the pegs with fibrinogen. And that's a
which
molecule released by the host when there's catheterization.
yeah, I was also thinking about, biofilms in more biology living tissues, like
Mhm.
it could be bones or other things. Is there any potential interest into looking how mutations would be different using, I don't know, there's a lot of material sciences that they are now 3D printing real tissues.
Yeah.
So would that be something possible to do in the future?
I think so. I mean, for one of these manuscripts that I talked about, we actually also looked at the adhesion to epitheliums. So in cell lines, I didn't show it here because it was
Mhm.
already too much. Um, and we saw that, again, the MRKD mutants, so the Fimbria mutants, bind a lot better to these types of epitheliums. But I mean, speaking from a clinical perspective, Klebsiella pneumonia is not often associated with biofilm formation in bones or stuff like that. So for this project in specific, I don't think it would be that relevant to look at biofilm in bones, but for other species like Staphylococcus, for instance, then definitely I think so. Um, but since Klebsiella is often associated with biofilm formation in medical devices like
Mhm.
catheters and so on, then it wouldn't be that relevant. But maybe for someone else looking with biofilms
Yeah,
formed by
that's
other
good.
species, then yes, it would totally be interested. And I think cotton was doing something like that with I think it was implants formed by I don't know, I don't remember. It was some
Some
material.
maybe bone or something. Yeah. Otilia, do you have a question? No. Okay. Let's see if anybody has any question. I have another one regarding antibiotics and the evolution experiment. So correct me if I'm wrong, but as I understand, you evolve the biofilms and then you explore how they can be treated either at high concentrations of antibiotics or also you're seeing that using sublethal,
Hmm.
these biofilms form on these already mutants that you selected. But have you thought to do a sub inhibitor concentration evolution experiment from the parentals before they can form biofilms? Because I'm thinking on a real scenario, right? These patients that get catheters, they are already at the hospital, they
Hmm.
might be already being treated with antibiotics at some degree, and then they get the catheters and they are there in the ICU for some time. So maybe looking at the evolution already with the presence of antibiotics. Is there something you've been looking at?
No, but it's actually a very good point. Because, like you said, during the clinics, the patients are usually exposed to antibiotics first, and then bacteria are forming biofilms. It's not something that we are looking at, and with the time that we have now, we we
Yeah.
won't do that. But it's a very, very good point, actually. So it's perhaps a follow-up.
For your next, your successor type of thing, maybe.
Yes, exactly.
All right. Anybody has any questions for Patricia? It was a wonderful presentation. I'm amazed at how much things there are on the things that you've been working on. Um, and actually, for a complex topic with a lot of different pathways, it's yeah, really good, very
Yeah,
interesting.
I try to simplify it because it's very, very complicated.
Sometimes it is difficult even to simplify something
Yeah.
like that, but good job. If nobody has any questions, I think we're gonna go to our break, like a leg stretcher, go get maybe some coffee, tea, gel out the window, maybe, and then come back. We will start with Otilia's presentation at quarter past.